Good day, everyone. Welcome to another class on pharmaceutical microbiology. My name is Bamidele Udumosu. In this class today, we will explore the cutting-edge field of biopharmaceuticals as well as the fight against the pernicious foe called tuberculosis. So we will look at biopharmaceuticals and antitubercular drugs on separate views. Let's go. So, what are biopharmaceuticals? Biopharmaceuticals are diverse medicinal therapeutic products that are obtained from biological sources and are produced specifically as a form of drug to treat infection. An example of biopharmaceutical includes vaccines, proteins, and other biological products such as blood components, somatic cells, gene therapies, and recombinant therapeutic cells. So, biopharmaceuticals are actually a way they are drugs that are not chemically synthesized. Most of the drugs available over the counter, sold in the hospitals, administered in hospitals, sold in pharmaceutical shops here and there, majority are chemically synthesized. Unlike that, biopharmaceuticals are products obtained or extracted from living cells and formulated into drugs. Biopharmaceutical can also be called biologics. That is, it consists of natural elements such as sugar, protein, or nucleic acids, or complex combination of these substances, and they may be living entities such as cells and tissues. Gene-based and cellular biology, for example, are often at the forefront of biomedical researches, and they are used in treatment of a variety of medical conditions of which no other treatment are available. So they can be obtained from natural sources, either from humans, animals, or microorganisms, and they may be produced by the use of biotechnology or other technological approach. Purpose is to ensure that they are provided and they are effective for treatment of ailments which other form of existing drugs are incapable of treating. So they can be produced from various techniques. For instance, recombinant DNA. This process is a well-known technology which involves the insertion of desired protein into host cell or desired genes into an organism to produce the same type of gene products in multiplied form or in accelerated form. In terms of protein, it is produced in a large quantity. We also have the type of production called hybridoma technology. Hybridoma technology is a well-established method for generating monoclonal antibodies which are highly specific, and they are pure molecules, and they are crucial in various biopharmaceutical applications. Hybridoma technology uses what we call tumor cells. It fuses the tumor cell with antibody producing B cells, which are usually isolated from immunized animals. So the resulting hybridoma cell line continuously produce identical monoclonal antibodies targeting a specific antigen, which is the immunizing agent. So the fused B cells with the tumor cell, which is also known as myeloma cells, help to create the epidermal cells and it produces the monoclonal antibodies. The advantage of epidermal technology is that it is highly specific. It can target specific antigen with minimized side effects. It's pure. It ensures consistent drug content and action, and normally have reduced allergic reaction. Then it can also be readily, cultured in cell lines for large scale production. And it has a tailored therapy in the sense that different antigen can actually be targeted, and it can enable personalized medical approach for various ailments. So hybridoma technology is one way of producing biopharmaceuticals. It can also be used in cancer therapy, infectious disease, such as those obtained from virus, bacteria, and all that. So the other method is also by cell culture, which is widely known again. That is the growing of cell in a controlled environment to produce therapeutic products from these specific cultures and be harvested and processed for administration into the recipient cells. So there are advantages of biopharmaceuticals, some of which I've mentioned earlier, in the sense that they have a targeted therapy. They can provide treatment for specific disease, unlike other drugs that may have side effects or are unspecific. Biopharmaceuticals have targeted therapies, so they are more precise and effective in treatment with less side effects. And they are also highly potent, active in lower dosage, better than traditional drugs that require continuous dosage regimen. Biopharmaceuticals are active in small dosage and they have improved efficacy than traditional drugs in the sense that they are efficient in treatment of certain diseases. They are highly efficient, unlike traditional drugs. And they also provide newer treatment options for diseases that have been regarded untreatable or difficult to treat, which might require long stay in the hospital. There are some cases that there are some diseases that have been found to be untreated before the introduction of these biopharmaceuticals. Some which are caused by microorganisms, some which are disease condition or malfunction. So biopharmaceuticals offer new treatment options in these cases. But we have challenges with biopharmaceuticals. These challenges are not actually disadvantages, it's just challenges. When we say challenges, they are the kind of barriers, hindrances. It's not really a disadvantage. So one of the challenges is that it's costly. Biopharmaceuticals are actually expensive than traditional drugs. In terms of production process, it also requires specialized expertise, and there are dedicated facilities that are required for production of these biopharmaceuticals, not found everywhere. So it makes it to be expensive. And also, after these biopharmaceuticals are produced, storage and handling are to be done with extra care. Any slight um, mishandling can result in spoilage because they are highly sensitive to temperature and other environmental factors. And one of the other challenges is that it's usually end up in limited supplies due to the high cost of production. Only few products are usually available. So these are part of the challenges associated with production and use and administration of biopharmaceuticals globally. Our next discussion is on antitubercular drugs. Now, tuberculosis, also known as TB, is a very dangerous infectious disease and caused by bacteria called Mycobacterium tuberculosis. Tuberculosis typically affects the lungs, but it can also spread to other parts of the body like the brain, spine, and kidney. It's a very dangerous infectious disease. Thankfully, TB is treatable, but it requires a multi-drug regimen and strict adherence to treatment plan. And this treatment plan depends on the type of TB infection. There's latent infection and active TB. Both required months of treatments. And if a patient can adhere to this, it will result in total eradication of the bacteria. If not, there could be complication because this organism has a high rate of mutation, slow growing bacteria, and it has the tendency of developing resistance to drugs. So therefore, treatment with TB has a peculiar way where we have first line, third line, up to fifth line of treatment drug. The drugs used in treatment of tuberculosis are classified into 5 groups, and this is based on their efficacy, evidence of their efficacy, potency, the drug class, and experience of use. So antitubercular drugs are actually based on these groups. So we will look at this drug and their mode of treatments in terms of line of treatment, one after the other. Now we have the first line of TB treatment. They are currently recommended in a 4-drug combination for the treatment of drug susceptible TB. These drugs need to be given in combination. There is monotherapy, but it will always result in resistance in most cases because of resistant strains. So in this context, a first-line drug refers to a medication that is considered the most effective and reliable choice for the initial treatment of TB. So they have been extensively tested and proven to be safe and highly effective against non-resistant strains. Now, non-resistant strains, because usually the first line treatment is expected to be able to eradicate these bacteria. Now, the characteristics of the first line treatment is that they are expected to be they are supposed to be potent, high efficacy, because they are expected to have a strong killing effect against a broad range of Mycobacterium tuberculosis strains, and they are expected to have low toxicity, and not expected to be expensive, and they are the well-established treatments. So these drugs include isoniazid, which is a primary bactericidal drugs that is mainly directed in killing M. tuberculosis bacteria. Then we also have rifampicin. Now, rifampicin is also a bactericidal drug with a fast-acting mechanism. It's bactericidal, and act so fast. Then we also have etabutor. Etabutor helps to prevent development of drug resistance and target specific component of the bacteria, such as bacterial cell wall. So, combination of this with rifapentine or rifabutine helps to eliminate this bacteria. Streptomycin used to be part of it, but over the time there has been resistance. So streptomycin in some cases are no longer included for this first line treatment. The second line drugs refers to those drugs that are used when the first line drug failed to deliver, when they are not effective against the infection. This usually occur when there is resistance. So the Mycobacterium tuberculosis bacteria may develop resistance and to render the first line drug ineffective. This can also occur when the complete treatment is not adhered to. So in the process of treating tuberculosis, the patient is expected to take the drug continuously. There shouldn't be a break. So if there's a break, sometimes this could lead to resistance. Apart from resistance, at times second line treatment is given because of adverse effects or intolerance by the system. In some cases, the patient may experience severe side effect or allergic reaction to first line treatment. So to make the first line treatment unsuitable for the treatment, then second line treatment is used. And in some cases, it could be that specific type of TB is detected. We have what we call extensive drug resistant TB. Example is Mycobacterium avium intracellulae. Now, this particular strain species is usually extensively resistant to treatment. So, first line treatment cannot be used for such. Rather, second line treatment is used. So three reasons where we can use second line is resistance, detected resistance, intolerance or adverse effect. Then we also have detection of resistant strains that require extensive treatment. So that will take us to the third line. The third line also refers to those drugs that are used after the first and second line treatment have also failed to deliver in terms of treatment, have failed to control the infection. This can also occur as a result of the first reasons given, such as resistance. And some cases too, we can have contraindications or intolerance or side effects, or can result in any form. This will lead to the use of the fourth line treatment. So the fourth line treatment is also a kind of recommended. They are not usually part of the first line treatment or second line treatment, but they are like the last resort option for cases where other lines of treatment have failed. So these typically occur in situations with drug resistant tuberculosis per se. Extensive drug resistant tuberculosis, apart from certain strains that are known to be extensive drug resistant, it could be as a result of acquisition of resistant gene by this tuberculosis along the line. Remember, the bacteria usually undergo mutation, which normally render this drug inefficient, especially the first and second line. So the fourth line drug is like the last resort option for cases of infection. So the group 5 third line antitubercular drug refers to the one classified as the World Health Organization Group 5 drug classification. It's like the fifth line drug. So it encompasses antituberculosis drug with unclear efficacy, meaning that there is no established enough evidence to confirm that they are efficient when compared to the first line or second line drugs, and they might be used against strains resistant to multiple resistant tuberculosis, or their optimal usage and effectiveness are still probably under investigation. So they are not usually given except under strict directive, and they are typically considered only when first line, second line have been regarded as failed treatment, especially when we have the MDR, drug resistant TB, or XDR TB, extensive drug resistant tuberculosis. So these typical group 5 are given in this category, but usually the fourth line is the last resort, which are those that are administered in cases where we have the multi-drug resistance and extensive drug resistance. However, the last fifth line, which is the World Health Organization Group 5 drug classification, are only given under certain directives and circumstances. And this consists of other different classes of antibiotics combined together for treatment. So that is all about antitubercular drugs, and I hope you have basic understanding on the first, second line treatment and third line treatment of these drugs for the treatment of tuberculosis. You have practice question just to see how you're doing, or how your reading can be applied on few questions for you after the slide.